Everything you need to know on the exam is in these 2 books, plus a few other things. So as I promised, DNA tumor viruses, RNA tumor viruses. Yeah, this is just the one lecture, and so that's all about the retroviruses here. So as usual, this is a review of what I think I did a bad job of explaining the first time around, and gives me a chance to do that and gives you a chance to ask me some more questions, if you have any at this point. And also reminds me, the exam, 8 o'clock Tuesday morning, yes, 50 multiple choice questions. Any questions come up between now and then, email me and I will try and both answer you and also post things on D2L. So if you don't want to ask a question, still check and see if people are asking questions there. So now we're talking DNA viruses and also these retroviruses way down here at the bottom, if I get my pointer to go that far. So we started out talking about the polyomaviruses and a couple of, I think, very important aspects about the polyomaviruses. This one is that we've got divergent promoters that we talked about last time, very similar to what you have with lambda. Also early promoters, late promoters, splicing that happens. All of these early genes are important for regulating the cell cycle and turning on replication. The late ones, again, not surprisingly, are all of our structural proteins. Important aspects about this, of course, is what's going on next to those promoters, and the big sort of take-home message with any kind of time you see a question on polyomavirus, first guess for your answer is T antigen. A particularly large T antigen. That might not always be right, but probably 90% of the time it will be, because T antigen basically does everything as far as the polyomaviruses are concerned. It stimulates transcription, it represses transcription, it has very important roles in replication, etc. So the other thing I wanted to mention here is this enhancer sequence, and the enhancers, again, originally found in SV40 in terms of cellular transcription factors or transcriptional activators, which are involved with completely normal promoters, TATA boxes, TATA binding protein, sort of you name it as far as that's concerned. Yes, large T antigen. Sure, I might throw it in as an outlier for some of these things, but for the most part, in terms of thinking about how these polyomaviruses work, I would say really focus on what's going on with large T antigen, because it basically, again, interacts with everything. Probably most important as far as the cell cycle is RB, and we're going to continue to talk about RB interactions with all of these polyoma, papilloma, adenoviruses, because that really seems to be mostly how they're driving the cell cycle. So binding to RB, which then allows progression to the cell cycle, which makes perfect sense if there are all these DNA viruses, because they need to have all of this machinery that the cell normally doesn't make, because it's not usually going through the cell cycle. Also gets into the nucleus, also true for all of these DNA viruses with one exception, pox viruses that we'll get to a little bit later on. So all of these things have to be in that place. I mentioned the J domain. This is something, again, one of those things I think I did a bad job of explaining before. This is a chaperonin-like domain, so it's important for rearrangement of structures of proteins, and it's particularly important with this rearrangement with RB. And so interactions with RB and the large T antigen interaction with RB as opposed to the normal cyclins, cyclin-dependent kinases, and other proteins which are blocking the activity of RB. And then our other friend as far as the cell cycle is concerned is P53. Normally P53 is our defense mechanism, so if you have a virus infection, you'll shut down the cell cycle and go into apoptosis. All of these double-stranded DNA viruses sort of take advantage of that and then have to tweak the P53, to make sure that the cells don't go into apoptosis, or at least don't go into apoptosis at a time that the virus, again, to over-anthropomorphize, doesn't want the cell to go into apoptosis. And then the last thing is this, as I mentioned, is a transcriptional activator. interacts with coactivators as well. And then finally, as if that wasn't enough for large T antigen, also acts as the replicative helicase for all of these polyomaviruses. So DNA goes down the middle, pulls the two strands apart, and those two strands being pulled apart, of course, is critical for DNA replication. Once you have the two strands that come apart, you can have all cellular replication machineries that come along with it, and it's this large T antigen which is most important for getting those two strands apart, again, in cells that are normally not undergoing replication. Papillomaviruses at a molecular level are very similar to the polyomaviruses. The structures are practically identical to each other. The genome arrangements are a little different, and how they end up stimulating the cell cycle is a little different. So wanted to mention that in a little bit more detail. Here, one thing that's quite different is instead of having lots of different splicing, which is what you have in the polyomaviruses, the papillomaviruses just have overlapping open reading frames. So for instance, the large, medium, and small T antigens in the polyomaviruses, all different due to different splicing. Here we just have different overlapping open reading frames. They're also all on one strand. You don't have a early versus a late divergent set of promoters. These are also splitting up this replication and transcriptional regulation. E1 basically is important for all of those replicative functions that you see in large T antigen. E2 is the transcriptional regulator that's involved in that. Of course, we also have to regulate the cell. That happens through RB and P53. Again, this is sort of an overview, a nice overview, in fact, of all of these different double-stranded DNA viruses and how they're interacting with RB and P53. So the papillomaviruses, E7 interacts with RB in an extremely similar way to the large T antigen, and as we'll talk about in just a second, E1A from adenovirus interacts with PRB to stimulate progression through the cell cycle. On the other hand, P53, the guardian of the genome, the protector, etc., normally, again, leads to blocking of the cell cycle, stimulation of apoptosis. Most viruses want to lower the activity of P53. The E6 protein in human papillomavirus lowers it, the large T antigen lowers it, and then E1B also lowers the activity of P53. So this is just, again, an overview, same sort of thing here. This could be E7, could be large T antigen, could be E1A. All of these, again, binding to RB, allowing E2Fs to lead to the rest of the cell cycle. P53 is particularly interesting in the papillomaviruses because E7 actually stimulates the activity of P53, which generally would be a really bad thing because that's stopping the cell cycle, it's leading to apoptosis, which is exactly the wrong thing that, again, to totally over-anthropomorphize, you want to happen if you are wanting to replicate as your virion. So E6 is sort of the yang to the yin of E7, and it causes degradation of P53. So you really have to have both E7 and E6 in order to get this proliferative growth. And also, as it turns out, this is a really potent way of getting cancerous growth because you're stimulating RB and then shutting down, P53. And that's why we get these DNA tumor viruses due to this overexpression of the cell cycle. And I talked about these really briefly at the end of the lecture, but I think it bears going into, and there was a question also on D2L about this, having to do with the different human papillomaviruses and how quite a few of them are involved in cancer. And so the real tumor viruses, these are the human papillomaviruses, it's mostly 16 and 18, which are the ones which are causing most cancers. But it turns out that there are a number of other human papillomaviruses which are causing cancer. This particular vaccine, the Gardasil vaccine, is, I think, if I remember correctly, the first recombinant vaccine. And so basically all they do is they make the major capsid protein, the VP1 protein, from all of these different HPVs. And they are different HPV proteins, and they all have slightly different antigenic determinants. But that's where you can basically put all these proteins together and make a really good vaccine. And the vaccine data that we'll talk about in just a second, when we talk about the pox viruses, don't include all of these cervical cancer reductions, but the amount of reduction in cervical cancer has been really amazing since the introduction of these vaccines. Probably wouldn't have HeLa cells if we'd had these vaccines a little bit earlier on. The last of these sort of DNA tumor viruses, even though adenoviruses really don't cause tumors in the vast majority of cases, are the adenoviruses. I am particularly fond of adenoviruses because they have these amazing structures that have been determined. This is the most recent one of those, literally this year, looking at the interactions of all of the multitude, actually, of capsid proteins. It's not just hexons and pentons. You also have various glue proteins that are holding these whole things together. These adenoviruses turn out to be really good at getting inside of cells and doing gene delivery. So a lot of the gene therapy processes, and in fact this particular virus is the basis for a lot of those gene therapy processes which are being used. Unlike the papilloma and polyoma viruses, these adenoviruses have linear genomes with proteins bound to the 5' end of each of those two strands. How do they get there? Because they're the protein primer for replication. So replication, polyoma, papilloma, all cellular with the viral helicase. Now it's a viral polymerase with a viral protein which is serving as the primer for your replication. The ends of these genomes have inverted terminal repeats, and if the book hadn't had a typo here, this really would be the inverted terminal repeat. CAT, CAT, CA at one end of the genome on one strand, the opposite end of the genome, CAT, CAT, CA. These are a little bit longer than just these, what is that, 7 nucleotides? 8 nucleotides, depending on if you have the typo or not. So this is going to vary from genome to genome of adenoviruses, but particularly these other viruses we'll talk about with these inverted terminal repeats sequences. Particularly the pox viruses have really long inverted terminal repeat structures. As we'll see in just a second, this is really important for the replication of the genome, particularly for getting extra strands thereof. And inverted terminal repeats are really good at doing what? Binding to each other. And that's what we'll see again and again in all of these other examples of that. So inverted terminal repeat and typo here. Binding and entry. This is a really nice figure that I keep harping on all the time. Not just for adenovirus, but also you can see here for herpesvirus, for HIV, SV40, parvoviruses. A really nice way of getting an overview of the entry processes that are going on here. Adenovirus has these projections, the 5-fold axis of symmetry, penton base, penton fiber, binds to receptors. You have receptor-mediated endocytosis. Then the rest of the capsid, after you've removed those pentons, gets transported to the nucleus. At the nucleus, the DNA is released and gets replicated. Herpes viruses, actually very, very similar. The nucleocapsid, because this one has an envelope on it, gets transported to the nucleus. There, the DNA is released. The parvoviruses, these are small enough that the whole virion can actually get inside the nucleus. The same thing is true with the polyoma viruses and also apparently the papilloma viruses, which can get into the nucleus. Those genomes, as I didn't mention before, are all packaged in nucleosomes, again, because this is all cellular processes which are going on there. And then finally up at the top here, HIV, again, is an enveloped virus with a nucleocapsid on the inside. That nucleocapsid is released and you have the reverse transcription of the genome that happens there, and then it's the genome which gets inside the cell. But let's back up again to the adenoviruses. Adenoviruses, big deal, splicing, 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 and that's because you have to use alternative splicing to get all of the late proteins. Late proteins also have alternative tailing. We end up with these multiple different classes of proteins, but even all the early proteins are spliced in different ways. Early proteins, E1A, E2A, these are the ones that are important for regulation of the cell cycle, but we also have DNA binding proteins, primers, this terminal protein, and DNA polymerases. So cellular polymerases, not just regulating the cell cycle, but also bringing in their own polymerases. Then, actually we'll skip over these guys. Classic, this is how we found about splicing in the first place. All these late genes are made from one promoter, has one leader sequence, and then gets spliced to all of these different ones. Then these short RNAs that I think I did a crummy job of explaining the first time around. These are virus-associated RNAs. That's what the VA stands for. But what they do is they block the activity of the protein kinase stimulated by double-stranded RNA. What the heck is that? PKR. PKR is one of the most important mechanisms, not just for RNA viruses, but also for DNA viruses. It turns on the interferon response. And the interferon response allows the organism to clear those viruses and does an extremely good job of it, partly through blocking translation, which I think we talked about it at some point. So that process and blocking that process is really important for getting virus replication. And so in this case, it's these RNAs which block the activity of PKR so that it doesn't lead to the stimulation of the immune response. Cell cycle stimulation, this should kind of sound a little bit like a broken record now. E1A, again, this could be large T antigen, could be various proteins from these other viruses. Bind to RB. Once they've bound to RB, the E2Fs can go ahead and stimulate the cell cycle. What is different about these guys, again, is the genome. They're double-stranded linear with proteins bound at the end. And so that means that the viral polymerase, cellular polymerases don't do very well with protein priming. So it's the viral polymerase that can bind to this terminal protein and extend from the OH, which is present on your terminal protein, extend your way down the genome. The single-stranded DNA binding protein, which is also a viral-specific protein, basically serves as the helicase. It binds so well to single-stranded DNA that it just pulls it apart in such a way that your polymerase can replicate its way down to the other end of the genome. In theory, you could just do this process again and again and again. This end, again, terminal protein, replicate it back in the other direction. In order to get lots of copies of the DNA genome, what seems to happen is these single strands can then also base pair with each other at these inverted terminal repeats. So remember, this sequence right here is identical to this sequence right here. That means that this sequence here on this strand is complementary to this sequence at this end. So each of those single strands can bind to itself base pairing wise, provide a template for the polymerase to extend its way around the genome. And this is how you can get exponential amplification of those adenovirus genomes. So now we're going to switch gears and talk a little bit about some of the guest lectures, just a few of the high points, I think, of some of those lectures, and the stuff that I think I understand relative to everything that Dr. Estep and pre-Dr. Kaysen understand. The structures, and we just talked about this just a second ago, of these herpes viruses, they've got a nucleocapsid on the inside, which is a pretty regular icosahedral capsid with a little portal protein at one of the five-fold axes of symmetry, extremely similar to bacteriophage here, the difference is they've all got these envelopes around the outside and proteins between this envelope and the nucleocapsid. And that's the tegument. This is this extra space here in the virion. And this has a lot of the important proteins that are needed very, very early on in virus replication. And the most important of these was VP16 that we'll talk about on the next slide. But these infection processes happen by binding of lots of different membrane glycoproteins to the outside and release of the nucleocapsid into the cell. Once the nucleocapsid in the cell, that nucleocapsid gets transported along microtubules. And again, many of these herpes viruses infect neurons, and these can be really massive cells. And okay, if you go on YouTube and just look for, herpes viruses being transported along microtubules, you can actually see some of these things, fluorescently labeled virions moving back and forth on the microtubules here. So here, move to the nucleus. At the nuclear membrane, the genome is released. You get DNA replication, transcription. That transcription is unfortunately named immediate early, followed by early, followed by late. Immediate early here basically means these are all of the genes which are going to be made by virus proteins that are coming in with the virion. So all stuff which is in the virion is going to be these immediate earlies. The earlies then get transcribed. Those then can lead to DNA replication. Once you've made DNA replication, then you can also express late proteins, which will lead to production of your structural proteins. The most important of these immediate earlies is the VP16 protein, present in the tegument as soon as it gets to the nucleus, serves as one of the strongest transcriptional activators that we know of. It's actually kind of interesting because VP16 in and of itself doesn't bind to DNA. It only binds to DNA together with cellular factors. These cellular factors then interact with the mediator complex, which allows the assembly of RNA polymerase II at all of the immediate early promoters. But this association between VP16 and the mediator is so strong that it basically steals all of the RNA polymerase II from any other gene and pops it onto those virus immediate early genes. What are those immediate early genes? These are lots and lots of them. The ones we mostly care about are ICP0 and ICP4. These are the main regulators of the early genes. So immediate early genes stimulate the production of early genes, and ICP4 also has a negative feedback loop which will block production of those immediate early genes after enough of them have been made to go to the early genes. So this is that process. I wouldn't bother about ICP22, wouldn't bother about ICP27. We've talked a lot about VP16. Now we've talked about ICP0 and ICP4. Replication in herpes virus, particularly in herpes simplex, which is the one that's best understood, is extremely similar to DNA replication that happens in Lambda and a number of other of these bacteriophages. Rolling circle replication, where one strand serves as a template, the other strand is being made continuously, and because it's being made continuously and there's a viral helicase that pull these two strands apart, you end up with many copies of the genome all attached to one another. And these are these head-to-tail concatenamers, just because the genome is being replicated around and around and around. Each of these segments, one genome at a time, gets cut and packaged into the nucleocapsid again, in a process extremely similar to what happens with the bacteriophage. Also, bacteriophages have scaffolding proteins that are important for making these icosahedral structures. Herpesvirus also has scaffolding proteins that are important for making these icosahedral structures. So there are many, many similarities with the nucleocapsid of herpesvirus and bacteriophages. One thing that's very different between the herpesvirus and bacteriophages is that bacteria don't have immune systems. And the herpesviruses, of course, are infecting organisms that do. And some of these, and the best understood are the, well, I'm not sure if Kaposi sarcoma is best understood, but that's what Dr. Estep works on, so he knows a lot about it. Epstein-Barr virus is a nice example, well, nice or bad example, depends on your point of view here. Another one of these tumor viruses. Epstein-Barr virus can cause a number of lymphomas. How does that happen? These are blood cell cancers. It's happening because a lot of the latent proteins that are present in EBV look like what would otherwise be a signaling protein that's present in B cells in the immune system. So this latent protein 1, latent protein 2A, both of these serve as signals for proliferation of cells that otherwise would not be undergoing proliferation. So B cell proliferation, and also one really good way that cells escape from being infected by viruses is to undergo apoptosis. Here, this is an anti-apoptotic function of these latent genes. Herpes simplex virus and that family, if I remember correctly, they're alpha herpes viruses, also have latency, so they hang out in nerve cells and occasionally get restimulated. There don't seem to be proteins that are important for that. It's mostly just transcripts, and so RNAs. And exactly how those RNAs are allowing herpes simplex virus to be latent is still a very active area of research, and no one completely understands exactly what's going on there. We know that they're being transcribed under latency, but how you get that induction coming from latency to virus production is really unclear at this time. And then the last of these herpes viruses, again, this is because I think to some extent it's what Dr. Estep works on, but also interesting aspects here. Again, I don't expect you to know all the details. I can't remember all of these details here. But it's mimicking normal cellular receptors. You've got viral proteins that are mimicking cellular receptors, basically, again, to stimulate the cell cycle. So these herpes viruses are undergoing this cell cycle stimulation in a very different way than the papillomas, the polyomas, and the adenos, which are all RB, RB, RB. Here, it's different signaling pathways which they're using to stimulate the cell cycle. And another thing about the herpes viruses, before I forget, is that these all seem to be, and we talked about this quite a bit when Dr. Estep was here, very species specific. So a simian herpes virus very rarely infects humans, and you've got herpes viruses that basically infect everything. And one of my favorite examples of that, one of my colleagues who's now down at Oregon State, had this really cool study. They were looking at corals. And the corals had these herpes viruses that were associated with them. And so, differences between humans and corals is a pretty large one. But all of these different organisms, and beluga whales, yes, also have herpes viruses that are associated with them. Are beluga whales going to be on the test? I don't know. The test isn't done yet. I've also got until Tuesday, just like you guys do. So our second guest lecture was pre-Dr. Kaysen. Talked a little bit about the single-stranded DNA viruses, particularly the ones that we're working on, or he's working on in my lab, but also some of the other single-stranded DNA viruses, which basically don't cause disease, but are potentially extremely useful for gene therapy purposes. So I mentioned adenovirus before as being one of the gene delivery vectors. Adeno-associated virus is even more heavily used, and in fact is even approved clinically for use in some gene therapy applications. So these are very small genomes with very few genes. Basically have a gene that's important for replication and a gene which is important for the structure. Splicing happens to give you different versions of these, but for the most part, it's replication at this end and the structural proteins down at this end of the genome. These now are completely dependent on cellular processes, but not even just dependent on cellular processes. Most of these viruses are the dependoviruses, which also depend on infection by a different virus. So adeno-associated virus in particular here, this guy down at the bottom, there's a whole bunch of different adeno-associated viruses, are dependent on an adenovirus infection of the same cell in order to get this virus replication because of the stimulation of the cell cycle that we've been kind of beating on here for the last half hour or so. But the replication of these genomes is very different. It's not double-stranded circular DNA, which uses cellular processes like our parvos and papillomas. It's not protein-primed replication. It's not rolling circle replication. It's genome-primed replication, which is a really fascinating process. So I wanted to look at that here. This depends completely on inverted terminal repeat structures that are also self-complementary, so secondary structures. And so this is kind of moving away from the DNA viruses. Remember all these RNA viruses, particularly the structures of the RNA which form, which are really important for translation. If you talk about irises, really important for regulation of gene production way back at the beginning, MS2. Yeah, I forgot about MS2. It was like forever ago. But it's a secondary structure, and these secondary structure formations are absolutely critical for getting replication to take place. And the reasoning for that is cellular polymerases. What do cellular DNA polymerases need? They need a template. They need a primer. So template is this single-stranded DNA. The primer is the 3' end of the genome. And it's base paired here in such a way that this DNA, once it's released by the virion in the nucleus, remember, because these all get transported to the nucleus, they're very tiny virions, now is a perfect template for cellular DNA polymerases. So cellular DNA polymerases will polymerize all the way to this end where it runs into a double-stranded piece of DNA. Cellular helicases will separate those strands. This gets copied all the way out to the end. Now you have this sort of funky structure, kind of like a handle at one end, with double-stranded DNA all the way down to the rest of the genome. This can now be transcribed into virus proteins. And so one of the important proteins, of course, that you need here is this rep protein. Sorry, Alice, I didn't see your hand.
Are the hairpin ends present in the virion?
So are the hairpin ends, is it paired in the virion? It does seem to be paired in the virion. And so that base pairing, that whole piece, so it's not as if it comes into the nucleus and then folds on itself. It seems to be folded up when it gets released. So as soon as it gets released, then the cellular DNA polymerases can extend them. Assuming those cellular DNA polymerases are around, which they won't be unless the cell's been pushed into S phase by some other stimulatory factor. Again, usually an adenovirus in this particular case. So first one of those viral proteins to be made is the rep protein. The rep protein is really an endonuclease. What it does is it cuts opposite where the 3' end of the genome was, now provides a 3' OH. The cellular polymerase can also extend out to the far end of the genome. Both ends of the genome now can form secondary structures, just like we had here. This can either be packaged directly as your new genome in your new virion, or can serve as a template for replication of more genomes. So this is that process for these parvoviruses. George also mentioned that they are important for gene therapy, not important in terms of human disease, but there are some veterinary diseases which are caused by these viruses, which are really pretty nasty, particularly dog parvoviruses, the canine parvoviruses, which can be really quite nasty. Fortunately, there are good vaccines for those. We work on single-stranded DNA viruses which have a circular genome, not those that have a linear genome with these funky ends to them. These just have a funky secondary structure which is present in the virion again as it's being released inside the cell. So now this is what happens with circoviruses and, again, probably our strange virus that we work with. These viruses, single strand except for these partially double-stranded pieces here, stem loop structure, again, complementary to each other. First thing that happens is you have a second strand which is made. How this happens is incredibly unclear for most of these circoviruses. Circoviruses don't cause diseases in humans, so who cares about them? And beak and feather disease virus. So if you like birds, it can be pretty nasty, but a lot less study's been done on these. So this first primer, not at all clear what's happening here. It may well be a cellular primer and the cellular primase. That's at least what happens in Phi-X174, which is that single-stranded DNA virus of bacteria that we talked about before. Once you, however, have double-stranded DNA, now you have the activity of a rep protein, which is incredibly similar to the rep protein that's present in these parvoviruses. It nicks the DNA here at this specific sequence, and then what it does is it takes the end of that DNA, so it's provided a 3' OH. What do you do with the 5' phosphate? You hook it up to an OH which is present on the protein. That's this connection that we have right here. The cellular polymerase, there should be a blob here. The blue stuff is all being made by the cellular polymerase. Helicase, sometimes the helicase is part of this rep protein, sometimes it's a separate helicase. This guy goes all the way around the genome, gets to the end here. Instead of doing rolling circle and making lots and lots of copies here, the rep protein reverses its activity, so it takes the 5' phosphate that was stuck to its tyrosine and hooks it back up to a 3' OH, and then you generate a single-stranded genome, and that can get packaged. So it's all about the rep protein. Binding, cutting, allowing a genome to replicate, religating that genome together, cutting, allowing another genome to be made, etc. That's what we think is happening with our hybrid virus or crucivirus. We have a circovirus-like rep protein that's present in our genome, but this very bizarre single-stranded RNA-like capsid protein that's also present in the genome. And we think that this RNA was stolen from an RNA virus, reverse transcribed and incorporated into this genome. How the heck that happens is what George's PhD is going to be about. So we think it has to do with the rep protein and this cutting and religating process, which could be doing this with RNA as well as with DNA. Okay, any questions particularly on those two lectures? Because again, those are not ones that I gave. I will be putting questions together based on the notes that I took when I was sitting back over there. Hey. No? Do you get cows that grow out of your arm after you get vaccinated with vaccinia? So, the Anti-Vaccine Society in 1812 was already telling you that you'll get cows that start to grow out of you, out of your nose, out of the side of your face, etc., once you get inoculated by these vaccines. But despite the cows, vaccines are amazing, really absolutely fabulous. And if we just, again, I like this figure in particular. If you look at morbidity in the US versus cases in the US, pre-vaccine, we're talking hundreds of thousands in some cases, measles. gone down to, unfortunately, now a little over 100, but just enormous reductions that have happened because of vaccines, and unfortunately these numbers are starting to climb back up again, not because of the anti-vaxxers, I just call them pro-disease, which I think is a much better way of talking about it. So there really should be the pro-disease society for its anti-vax societies. Okay, so we talked about vaccines in the poxvirus lecture because vaccines, particularly the vaccine against variola, the one that made cows grow out of your arms, is the reason that we don't have smallpox anymore. So what does that genome look like? Turns out that genome is quite similar in many ways to the really tiny genomes, the parvovirus genomes, even though these are some of the largest genomes, about 200 kilobase pairs in size. They have these ends which are looped around. It's actually one contiguous DNA molecule, 200,000+ base pairs in length. Again, linked together at one end. Also have these terminal repeat structures which are mostly self-complementary to each other. Not surprisingly, replication seems to happen by cutting and giving a 3' OH that can then be extended from there. The big difference, of course, between the parvoviruses and the poxviruses is that the poxviruses are big. They've got a whole bunch of proteins. They're encoding lots of proteins. Their virions are very large. They have a bunch of proteins that are present in their virions. And so much, in fact, that these viruses can replicate completely in the cytoplasm, independently of the nucleus, except for a few of the products, which again, the deoxyribonucleotides and some of the other things. But all the proteins, the DNA polymerases, the RNA polymerases, all of these are either encoded in the poxvirus genomes, or all of these early genes are packaged in the virion. So somebody asked me, I think before, maybe it was after class, why don't you call this the tegument between the membrane on the outside and the core of the virus on the inside? The reason for that seems to be that poxvirologists don't talk to herpes virologists. So the concept is extremely similar. Again, these are proteins that are packaged between a membrane and a core structure, which are then active as soon as they get released inside the cell. So these proteins lead to production of, also they call them intermediate ones here. I would call them early proteins. So these early proteins include intermediate regulators. Intermediate regulators lead to production of late regulators. These late regulators then get packaged as proteins in the tegument equivalent in the virions, which then get released and can go back and go through this whole structure. This process, now it's shown here as being these big lines going around the outside. The virions aren't moving around like that. They're actually all present in one big agglomeration outside of the nucleus, and that's the virus factory, where you see all of the viral proteins, where you see all of the viral nucleic acid in one almost separate organelle that you see inside these infected cells. Replication, as I mentioned before, is very similar conceptually to parvovirus replication. You have this one contiguous nucleotide structure, again, one molecule gets cut at a particular position. That provides a 3' OH and a template, which extends to the end. This would be a problem if you didn't have complementarity in order to form these secondary structures, which will then provide 3' OHs and templates, so you can make multiple copies of these genomes. Unlike the case with the parvoviruses, these are linear, you will fall off the end here. These are circular, so you end up with concatameric structures, so multiple genomes hooked up next to each other. Those then have to be cut and packaged into individual genomes, which then get packaged. That Concatemer resolution process actually seems to be similar to what happens in homologous recombination, where you have crossing over and holiday junctions, which form. Those proteins are actually very, very similar to each other in structure. We mentioned a little bit, and I got to this only right at the end of the poxvirus lecture, so I wanted to mention a little bit more detail here. There really is kind of two sets of virions that get formed in poxvirus infections. There's the really stable version, which is down here, that just has one membrane on it, and the unstable version that can go and infect other cells that has multiple membranes around it. And this is the process on how that happens. So here you have production. Again, this is all happening in the cytoplasm. Gets its membranes and the membrane proteins from the Golgi apparatus. And so now we've got this mature virion here with these proteins around the outside. So the red dots right here. And this buds into a intracellular compartment. This then can be released. You'll have one single membrane around the outside with those specific membrane proteins that are in it that are really important for infecting other organisms. On the other hand, this guy can get another set of membranes added around it, which now have different membrane proteins in them. This then can fuse, and you'll end up with a second set of membrane proteins here, and these are the ones that are important for cell-to-cell infection that you see, and the rockets that I talked about. So these also will polymerize actin, and that actin polymerization will literally push the virions out of the cell and into nearby cells. Yeah?
Sorry, just clarification. So is the secondary envelope really Golgi yet again?
Yeah, it seems to be that it's a Golgi-derived membrane as well. And it makes sense because these are membrane proteins. Membrane proteins are going to be made by the ribosomes, which are going to be associated with the Golgi in the endoplasmic reticulum, because that's where all these membrane proteins are being made. Yeah?
And the mature virion is the one that infects other organisms?
Yeah, well, so there's the intracellular mature, which is infecting other organisms, and the extracellular mature, which has that extra membrane. That's going from cell to cell.
Okay, gotcha.
Yeah, they're both mature then. Gotcha. Yeah, so, but it's the IMV and the EMV. Yep.
Is it acquiring different types of membranes in the Golgi? Like, is there a different composition to the early?
Oh, the two different membranes, are they different in terms of their lipid composition? That's a great question. I do not know the answer to that. Certainly the proteins are different, but whether the lipid components are different and exactly how it's picking those up, that I don't know. It may well be in this textbook, which is hopefully still on reserve. Anybody go to try and find it?
Yes.
Yes, it's there. Good. So that would hopefully have some more details on that. But I don't know. If I don't know, highly unlikely to be on an exam. So now let's talk about this book in, 4 minutes. So these are the retroviruses. Classic retrovirus. The genome is basically a messenger RNA with a cap and a poly A tail with a repeated sequence, unique sequences at either end, 5' and 3', and then these 3 sets of genes gag the group-specific antigen. Most of these are the structural, particularly the nucleocapsid proteins, the polymerase genes, which are all of the enzymes, not just the reverse transcriptase, and then the envelope proteins. These are the ones which are going to be sticking out in the envelope. Again, that Flint textbook here, I'm vaguely thinking about switching to that textbook in a couple of years. We'll see. May get there when that happens. Binding to receptors on the outside of the cell. The nucleocapsid is released into the cytoplasm. Here, the reverse transcriptase has its activity. So the reverse transcriptase in the cytoplasm will replicate the genome and take that RNA and convert it into double-stranded DNA that we'll look at in more detail on the next slide. Once you have double-stranded DNA, this then needs to get into the nucleus in order to have integration to take place. For the lentiviruses, the HIVs, this is done actively. Most retroviruses, this actually happens passively, and they can only replicate after you have cell division. And so the nuclear membrane breaks down because this intosome, as they call it, which is the double-stranded DNA copy with some extra bits on the end of your retrovirus genome, plus the integrase, is too big generally to get into the nucleus just by itself. Once you're integrated into the host genome, this gets transcribed by cellular machinery, translated by cellular machinery. The translation, you end up with mostly structural proteins, but occasionally you'll have a frame shift or translational termination suppression and make this polyprotein, which includes all of the enzymatic proteins. So GAG-POL, polyprotein. This gets targeted to the membrane, where you also have the envelope proteins. The envelope proteins are all made from a major splice product, which takes out the GAG-POL genes and just allows the envelope proteins to be made. These are made as normal envelope proteins, ER getting translated to Golgi. Those then in vesicles get transported to the plasma membrane. They all associate here with 2 copies of the genome. Those then, these particles, then get assembled, and only at this very last step do you have proteolysis that takes place, which chops up the GAG and the POL into separate proteins and allows them to be active. So it's a post-budding process, which is absolutely critical to get infectious retroviruses. Yeah?
Might not be relevant. Do you think that this happens, or that it's evolved to happen because if this kind of process was taking part in the cell before you package them in a virion, you might lose some proteins, or things might get scattered, or you might trigger a host cell response or something like that?
Yeah, so the question is, again, why? Evolutionarily, it works. But it certainly does make sense that you don't want to have all of these enzymes being active in the cytoplasm, because proteases chopping up all kinds of things. You've got activity of reverse transcriptases might bind to cellular RNAs and make copies of those. All kinds of possibilities that could be happening. So it makes sense to have the activation of your enzymes in particular happen extracellularly. So, but again, how that could have evolved, I was just trying to think about this on my way in today. It's like, wow, how the heck could something like this have evolved? This is just, yeah, crazy. So, don't think it'll keep me up nights, but we shall see. So the replication process, what happens with these genomes? And again, this is a question on how the heck did something like this evolve? So you have your genome. Your genome is always base paired in the virion with a cellular tRNA. That cellular tRNA is the primer for replication of the negative strand. And sorry that a bunch of people missed that clicker question. So negative strand versus positive strand. Positive strand's the genome. That's what codes for all of your proteins. But unlike the RNA viruses, this doesn't get translated directly. It only gets translated after messenger RNAs are made a lot later on. So the negative strand is made starting with this tRNA, which serves a primer, bound to the primer binding site. So tRNA, RNA primer, RNA template. Here, the reverse transcriptase, which is a DNA-dependent DNA polymerase for later steps, but now it's an RNA-dependent DNA polymerase, classic reverse transcriptase. RNA makes a DNA copy of it, will make a copy out to the end of the genome. And this repeated sequence, which is present at the very end of the genome. This RNA-DNA hybrid is not stable. RNase H's. RNase H would normally be what's happening in the nucleus to get rid of those extra Okazaki fragment primers, etc. This again, however, is happening in the cytoplasm. So the RNase H here is part of excuse me, the reverse transcriptase. So that RNase H will chop up the RNA in this RNA-DNA hybrid. And this is still negative strand here. It's also, they call this the negative strand strong stop, because you see a lot of this particular small piece of DNA just from the primer binding site out to the end of the genome. This repeated sequence, of course, is repeated at the opposite end of the genome. So you have strand transfer that takes place, again, either the other copy of the genome, because there are always two copies of genomes that are present in these retroviruses, to the opposite end of the genome. Now it's base paired over here. Now I've got a 3' OH. We've got a template. We're good to go. We can replicate all the way down to the opposite end of the genome. Again, this is your negative strand, which is being made here. So all the negative strands all being primed by this tRNA. Once you have this whole piece, again, it's an RNA-DNA hybrid. RNase H can chew this up. This polypurine tract here, PPT, turns out to be resistant to being degraded by the RNase H. And that means you end up with a small amount of RNA, which is still here, which can serve as a primer for the positive strand of the reverse transcriptase. So it will bind here, extend through this tRNA until it gets to the end of the primer binding site, which is a pseudouridine. It's one of those modified bases present in tRNAs that the polymerase can't do anything with. So gets there, it stops. The RNase H finally chews up the PPT and the tRNA. And now we've just got a partially double-stranded piece of DNA with a primer binding site here. Again, this is positive strand. The negative strand has a complement of that. So this guy, again, can either loop around and copy at this end or shift over to a copy which is being made by the other strand, the other polymerase. And you have 3' OH, a template, 3' OH, a template. This gets replicated again, all in the cytoplasm, all by the reverse transcriptase, giving you this long terminal repeat at either end, which has that repeated sequence that you started with over here, but has also added the unique sequence from the 3' end at the 5' end, and the unique sequence from the 5' end at the 3' end. Yeah?
So sorry if I missed this. Is this circularized?
So here, this piece is not circularized. This is a linear piece. But you could have circularization that takes place between this end and that end. But you're always going to end up with a linear piece of DNA which has these LTRs at either end. Those then bind to the integrase, which will take it into the nucleus and pop it into the genome. Yeah, Alice?
How is the double-stranded DNA protected in the cytoplasm, or is it just like timing nuclease so that you're not...
So how is it protected? So the nucleases don't break it down. It seems to be still in the core. And so it's actually protected by those proteins in the core. So it seems to be mostly protected due to that. Once, however, you've made this double-stranded DNA, the ends are bound by the integrase. And double-stranded DNA just by itself, you don't have too many endonucleases floating around in the cell. Endonucleases would be really, really dangerous if you have endonucleases floating around. Exonucleases are all over the place. So as long as you protect ends, you're in pretty good shape. So once you've done all of this, now single-stranded DNA by itself is incredibly mutagenic and all kinds of other things. So everything up to this last step you want to have happening in the core. But once it's done and once you have the integrase bound to the ends, that's a pretty stable intosome which can then be incorporated. Really briefly on HIV, again, you could have a whole course on HIV. We did have a course on HIV a couple of years ago. The things to mention here, these are all exactly the same things. Cool structure of the nuclear capsid. Instead of having a icosahedral nucleocapsid structure, it's got one that looks like half of an SSV in here. So that we think is really quite interesting and maybe a new sort of paradigm for virus structures. There's a lot more splicing that goes on in the HIV genome, generating this TAT and REV protein, which are not present in most retroviruses. The TAT is this transactivator of transcription, which serves as an anti-terminator, functionally pretty similar to what happens in lambda with Q and N, structurally completely different, but just conceptually the same. REV is very important for transporting unspliced RNA out of the nucleus so that it can get packaged into new virions. So those are the only ones that we really covered in any detail of these extra proteins that are present in HIV. Take a whole course, all kinds of other interesting things going on there. So giant viruses, the main thing about these giant viruses really is the translation machinery which is coded for in their genomes. If you look at PBCV1, it's got a bunch of tRNAs that are present in the genome. Otherwise, the genome is extremely similar to the pox viruses. Its infection process is a lot like bacteriophage, probably because these are infecting algae that have really tough cell walls. So not unlike the bacteriophage. Bacteriophage have to get inside the bacteria that have really tough cell walls. Here, they've got to get inside the algae, which have tough cell walls. So this binding, degradation of cell wall, release of genome that goes on the inside. We talked really briefly about the EHUX viruses that change the weather just because of the massive numbers that you have of the coccolithophores and the coccolithophore viruses, which are infecting things that are in there. But genome-wise, Mimivirus was the king. Now we're at Pandora virus, which is twice as big, but really, really, really massive genomes. This is 1 million base pairs in size. Has, not unlike the pox viruses, which are related to these giant viruses, DNA repair, but mostly importantly, the translation machinery. None of the other ones until they found this one had aminoacyl tRNA synthetases as well as tRNA. Also have the cap binding protein, so this EIF4E homologue. So basically everything but the ribosome as far as we're concerned for all of these viruses. They replicate again through virus factories outside of the nucleus, but very large amounts of production of viral nucleic acid and viral proteins. These megaviruses, the giant viruses, have viruses that can infect cells that are infected by them. And so I like to think of the virus factory as kind of like being what's being infected by these virophage. These are the Sputnik and Ma viruses. The Ma viruses in particular are very similar to transposons. So transposons versus viruses, the difference between these two just seems to be you got an extra capsid protein. Capsid protein plus transposon basically gives you a virus. Okay, so what does that mean about these various different viruses? So viruses not having ribosomes, okay, that's one definition of your viruses, or virus is just a mobile piece of DNA with a capsid protein on it. Okay, so two real extremes in studying these extremophile, let's say extremophile viruses. No, the extremely large viruses. The extremophile viruses are totally cool and the most important and interesting viruses ever, but we don't have any more time to talk about them today. Any more questions that come up, again, please email me. Enjoy your weekend if you can. It's going to be rainy anyway. Thanks for a wonderful term. Greatly appreciate it. Yeah, so I'm sorry. All my exams are in theory just since the last exam, but I will assume that you know information from before. So I won't specifically have questions on things before that, but I will assume that you know some of that. Kind of the stuff like we're talking about today. Similarities to MS2, similarities to 5x174, those kinds of things.